Welcome to the Huck Distinguished Lecture Series. This is the second one of the academic year, and we're very much looking forward to the talk today. My name is Laura Weirick. I'll be moderating the session. I am a co-hired faculty member between the Huck and the Department of Anthropology. And I just want to take a second to say thank you to all the Huck staff, and especially Melissa Bailey, who helped organize the talk today. We will go ahead and have an introduction today by Ileana Boms, and then we will go ahead with the talk. Immediately following the talk, we will have a question and answer session. If you would like to ask a question, please use the blue raise your hand function in the participants tab, or you can feel free to type it into the chat and I will go ahead and read it out. So with that, I would just like to take a moment to remind you all to keep your mute on so that we don't disrupt the talk, and encourage you to turn on your video if you're able. And with that, I will allow Dr. Boms to do the introduction.
Hello, everyone. Thank you, Laura. And it is my great pleasure today to introduce Professor Dr. Thorsten Reusch. I first got to know Thorsten when I was planning my PhD research on coral clonal reproduction. And I was looking for a sampling design that would allow me to detect these coral clones. And the best protocol I could find was actually for seagrasses, published by Thorsten's group. And ever since, I have followed Thorsten's research with great interest, and I've visited him in Kiel a couple of times. And so today I'm very happy to welcome Thorsten to Penn State, even though it's remote. Thorsten is the head of the Helmholtz Center for Ocean Research in Kiel for Research Division 3, Marine Ecology, since 2012. He's also a member of the executive board of the Excellence Cluster of the Future Ocean since 2011. From 2005 to 2008, he was the managing director of the Institute for Evolution and Biodiversity at the University of Münster in Germany. And he was a Heisenberg Fellow for the Deutsche Forschungsgemeinschaft and a Marie Curie Fellow at the University of Groningen. Before that, he postdoced at the Coastal Marine Institute in San Diego, and he obtained his PhD at the University of Kiel in 1994. He's an associate editor for the journal Marine Biology. Dr. Reusch's research includes evolutionary biology and population genetics, host-parasite interactions, and evolutionary consequences of global change. His research focuses on seagrasses and fish, and he frequently uses the Baltic Sea as his natural laboratory. He says he sees any ecological interactions as selection pressure, and consequently, if there's heritable variation, evolutionary change will take place. And this focus on connecting ecology and evolution has produced some really novel insights, and it is therefore no surprise that his group frequently publishes in top-tier journals such as Nature, Global Change Biology, Current Biology, and Trends in Ecology and Evolution. So please help me welcome Thorsten to PSU. We're very much looking forward to your talk.
Yeah, thanks a lot, Jana, for the invitation, for the nice introductory words, and also thanks to the Huck team to make everything possible here. Yeah, so let me start the lecture right away. Just scream and yell if it's not in the full screen mode at the moment, but we tried it 10 minutes ago. Yeah, so I would like to introduce a marine evolutionary time machine to you. And actually, Ileana already did the spoiler and told you that it is the Baltic Sea, which is often our evolutionary lab. But anyway, that's just fine. So some more brief words on GEOMAR Kiel. What is that? So GEOMAR is a research institution that's operating in collaboration with the university, and all faculty are full members of Kiel University, but otherwise the budget is independent, it is federal. So we have quite considerable budget and about 1,000 staff, students, and faculty. And yeah, so we could be called the German Ocean Institute. My group in particular, as Ileana nicely summarized already, sees natural selection as ecology in action, which is precisely another definition of evolutionary ecology. And just to remind some of you that may not be so familiar with evolution concept, so evolutionary adaptation is not acclimation. So tanning would be a form of reversible phenotypic plasticity that adapts you, of course, to increasing solar radiation, in particular protects you in the face of UVB, but this is not evolutionary adaptation. That's a population level process. Another example for irreversible plasticity during ontogeny would be those spines of Daphnia, of water fleas, that protect them against fish predation, but only in those ponds that contain fish. So that's also reacting to certain odor smells. Evolutionary adaptation, where we'll give you a number of examples from the Baltic Sea later. The general concepts were already foreseen by Darwin, although he had no idea whatsoever how the genetic heritability itself would take place, but he just postulated that there would be heritability among individuals in certain traits. And under global change conditions, we also have a changing or a novel environments. And some individuals, owing to those traits, have a higher reproductive success, or we also call that higher Darwinian fitness. They are then positively selected, and that means that the population mean fitness increases. And if you look then at population genetics or genomics, that would mean ultimately that allele frequencies have to change across spatial locations or over time. So far, so good. Just this really quick primer on evolutionary principles. Now let's switch back to marine ecology and the influence of humans on marine ecosystems. Here it's very illustrative to bear in mind that most of the human population is living very close to the ocean. So it's really this interface between the open ocean and the coastlines where the human influence sphere meets the marine ecosystems, and therefore where the pressures are also most felt by marine ecosystems. And here, of course, it's also no surprise that some semi-enclosed seas, here the Baltic Sea, that's at the eastern edge of the North Atlantic, they of course feel human anthropogenic influences more than other world regions. So in the Baltic Sea region, here just to make sure that you get the geography right. Can you see my cursor actually? Can anyone confirm that you do see my cursor? Okay, let's just hope that you do see my cursor. So this would be Germany. Sorry?
We can see it.
Okay, great. And here would be Russia, right? That would be Scandinavia. So you see here that this ocean area is surrounded by 85 million inhabitants of quite developed societies. So there's a lot of industrial influence, but also of intense agriculture. And the mean depth of that ocean basin is only 85 meters, meaning that the water volume is relatively low and the exchange time of an average water particle with the open Atlantic is 80 years. Surrounding the Baltic are many regions, many countries where there's a lot of scientific activity going on, and there are also a lot of European Union programs particularly focused on that Baltic Sea region, one of which in the past 8 years was the BONUS program. So that's where at least 3 different nations had to come together with a joint research program, and that program was very successful in that it promoted the Baltic Sea as a time machine for the global coastal ocean. So if you will, that was an exercise of a network of already networked projects. So 8 of those EU bonus projects got together under our coordination. We had 2 longer writing team sessions that you see here. And the eventual outcome was that we tried to promote the Baltic Sea as a time machine for the future coastal ocean. Of course, there are some parallels that will not work. So there's never, of course, a template for each and every world region, but I will now walk you through some of those aspects where we think that the Baltic Sea can give you some messages for other world regions. Let's first turn our attention to the environmental impacts, to the factors that are rapidly changing under global change. One of which you see here, that's the degree of warming per decade in Kelvin. And you do see that in particular, the central and the northern Baltic up here, they warm with about triple the speed than the average North Atlantic. Yes, there are also other regions like the Black Sea or the Eastern Mediterranean that are warming quickly, but this is already a quite drastic signal. The Baltic is even more unique in its acidification levels. So depicted here, over one season in the year 2015 are the PCO2 values, just to compare. So the atmosphere right now is at about 420 parts per million partial pressure of CO2. And this is what we see in the Baltic Sea. And here, this is the ocean of the year 2010, the range that is predicted under different carbon emission scenarios. So now you might ask yourself, so why is the Baltic Sea already experiencing now such high PCO2 values that are even exceeding the ocean of 2100? The reason is that there's a lot of oxygen depletion, and that oxygen is converted into CO2 and is adding to the acidification levels that come through the dissolution or the equilibrium with the atmosphere. And we can also make a more formal comparison here. So these are mostly US American long-term monitoring sites of the PCO2 value here in Puget Sound, Alaska, California, Florida, Hawaii. And you do see that, again, the Western Baltic Sea sites are right on the doorsteps of our institute. They really stand out in terms of the mean values and the fluctuation. And what kind of evolutionary consequences this can have, I will come in the second half of my talk. But to sum up here, so the Baltic Sea in a time machine in terms of warming and of acidification, everything plays out in fast forward. Another very important anthropogenic impact is, of course, overfishing. And here you might think that the fish stocks are quite well managed, right, in the on the doorsteps between Sweden, Poland, Germany, and Denmark, but this is clearly not the case. So one of the stocks that really gives us a lot of headache at the moment, provokes a lot of sorrows, and also has severe socioeconomic consequences, the decline of the Western Baltic cod stock, a relatively small stock separated from the Central Baltic cod stock, but nevertheless very important regionally. You see here, it's just down a slippery slope, if you will. I would also like to pay attention to the yellow bars here. That's the recreational catch by anglers. And because the professional landings have become so small, we now see about a third of the catch by anglers. So that tells you in which miserable state the Western Baltic cod stock is. This is not just bad for the socioeconomy, but it also results, and there's now really strong experimental evidence, it results in trophic cascades. So the natural state, if you will, is large cod keep smaller coastal benthic fish in check, like the stickleback, or also pipe fishes, which then cannot as easily consume mesograzers, such as isopods or also snails, and they then keep macrophytes clean and happy so that they can reach the light and that they can proliferate. The trophic cascade that now has ensued in many areas, in particular of the Western Baltic, is that the small coastal fish have increased tremendously. So there's actually a stickleback wave. There's just been, I think, a Nature Communications paper on that, because they're not limited anymore. They then decimate the mesograzers, and the result is macrophytes overgrown with opportunistic algae and a loss of this very important and critical nursery habitat. So to sum up, the Baltic is a really good, bad example, if you will, for multiple pressures that exceed those pressures in many other world regions. So here we tried to make a semi-quantitative comparison to the North Sea, to the Mediterranean Sea, Black Sea, Gulf of Mexico, East China Sea, and the Barents Sea. The Baltic Sea is not the worst everywhere, but if you look really at the aggregated amount and the diversity of pressures, it has been for decades now in a really bad shape. But that's only half of the story. The good story, this has now nothing to do with evolutionary biology, but I think it's important to mention in times where international collaboration is sometimes not so much valued, is that it has also, since a number of decades, a quite well-functioning governance regime and monitoring and research regime, if you factor in that there are 9 different countries neighboring it of 2 very different political blocs. So there's Russia and there is the EU. So for one thing, I just have mentioned that bonus programs. So they're very strong research activities that have as a condition that you team up with at least 2 other countries. And there's also a wealth of long-term monitoring data available that are very essential, not only to figure out that there is some problem, like with the cod stocks I've just shown you, but also to figure out if you do a positive intervention, for example, if you try to curb nutrients to fight eutrophication, that there's also an improvement. Without long-term monitoring data, if you cannot show there's an improvement, it's of course very hard to conduct a science-based management. And actually the precursor of all that lay the foundation of that long-term collaboration was the International Council for the Exploration of the Sea. So that was founded already in 1902 by, I think, exclusively male human beings here that had to have a beard and a black hat. Okay, so there are, of course, sociological reasons why at that time these were mostly males, but why they had to have a black beard and a black hat, that escapes my knowledge. But nevertheless, it was very useful that they started that completely impartial and non-political scientific collaboration. And that was the precursor to have really good monitoring data and also scientific collaboration throughout the entire North Atlantic area, not just the Baltic Sea. Now, some very few words, final words on the governance structure. That's also very peculiar. I had already started mentioning that. So here the Helsinki Convention, that's one of the first regional sea conventions already formed in 1974, when the status of the Baltic Sea, owing to these many pressures, was in a pretty abysmal state. So the Helsinki Commission was founded, and the signatories at that time were all of the neighboring countries, also the Baltic state that at that time were still belonging to the East Bloc, so were members of the Soviet Union. And this HELCOM survived all of the very partially heavy political transitions, kept on going, planning ambitious environmental plans. For example, the Baltic Sea Action Plan for Sustainable Management of the Baltic Sea that was endorsed by all member states in 2008. In 2008, as you may or may not know, the Baltic states now had already become members of the EU, and so did Poland. And the Soviet Union mutated into Russia, but still Russia is kept on board with those plans, because that's not just the EU running that, but the HELCOM. Excuse me. So what are the management successes? For example, a really big management successes is the return of top predators, very important, of course, for top-down regulation of ecosystems. It's, for example, the return of the wild salmon. This is partially a success of recognizing that they are genetically distinct populations roaming in the different tributaries, so that you cannot just bulk restore with fry from anywhere else. So that was a very important science message here. And also there was some successful nutrient reduction. I'm saying some here because you may see that the peak nutrients were in the 1980s, 1990s. Then nutrient pollution was reduced, but then the reduction kind of stagnated. We think that's because all the low-hanging fruits have been picked, and also because warming is now eating up. So warming and increasing decomposition, increasing microbial respiration is now eating up the initial management successes. But now let me come, after I've illustrated you the selection regime in the Baltic and its enormous temporal dynamics, let me now introduce you to the Baltic as an evolutionary time machine. And here we have to deal with a paradox. So we have, in a way, a dead end of marine species that are also confined to a relatively limited space compared to the vast areas of the Atlantic. So we would immediately predict we have smaller population sizes. We have a rather stressful regime with lower salinity levels, progressively becoming almost freshwater in the top north pocket. And so therefore it's absolutely predicted that all the molecular marker data that you can find for pairs of populations of the same species in the Atlantic versus the Baltic, that most of those pairwise comparisons are below the equal line x equals y equals z. And the extreme is, of course, that there are many populations that are really predominantly clonal in the mid and the North Baltic. So they are not featuring any diversity at the level of sexually reproducing individuals. So Fucus radicans, for example, that's a Fucus species that's entirely reproducing asexual, and the seagrasses that Iliana had already mentioned are also very famous examples of forming clones that are several football fields large, maybe even larger, for example, in the Finnish archipelago. However, just to mention that here briefly, even clones may be able to evolve. So some new findings from our lab have shown that there are many somatic mutations arising seemingly in a seemingly very small proliferating tissue of those macrophytes, and they are then distributed into a fixed mode among the ramets, among the modules of such a seagrass clone. So in principle, adaptive evolution would be possible here. And I'm very happy that Iliana Baums, Benjamin Werner, and myself, we got a Human Frontiers of Science project called Adapt Asex to observe and study exactly these processes in corals, in seagrasses, and in cancer. Let me now go through some of those environmental factors that can elicit evolutionary responses, one of which that's rather overlooked in marine systems is actually salinity change. In many ocean basins, either owing to enhanced evaporation in the tropics or enhanced runoff and or melting of glaciers, salinity changes are expected in the surface waters. And the Baltic here lends itself as an ideal space for time design to have a look how originally marine organisms, such as the three-spined stickleback, that by origin is marine, it just entered freshwater secondarily, how they cope with a gradient of salinities. So you see here the Baltic in these different colors, and the salinity goes from fully marine to almost freshwater, here the bluish colors, in the Bothnian Bay and in the Finnish Bay. So we took here 3 different sites that were also already studied by De Favre and Merille some years ago that do show clear local adaptation in terms of various responses. So in particular, under high salinity here on the left-hand side, you do see that the freshwater adapted populations really decline in the body size, whereas those from almost marine conditions, they are actually adapted to all 3 different salinities. So there's a quite strong local adaptation aspect to salinity, but the genomic differentiation overall is very, very low. So this is now based on full genome resequencing. And we're now asking if the very much hyped epigenetic pathway may contribute to phenotypic adaptation in sticklebacks. And so according to this scheme, DNA methylation variation sits kind of in the middle of that Darwinian cycle, if you will, that where normally genetic variation, either standing variation or owing to new mutations, underlies phenotypic variation. But what has been overlooked so far, in most cases, is that there are 2 sorts of methylation variation, if you will. So one is what we call stable. Of course, it's not stable in evolutionary times, but it's stable at least for experimental generations. That's a methylation mechanism that occurs like random normal genetic mutations and alters the methylation patterns and hence gene expression and various traits, or it's inducible. And then this is often equated as adaptive in the sense of adaptive phenotypic plasticity. So you put an individual in a new condition and then in a regulated way, new methylation sites appear that would then lead to a better adapted phenotype. And clearly those pathways are, in terms of the genesis and their consequences, very, very different. And now we wanted to find out, again, in one of those bonus EU projects called Bambi, nicely led by Cash and Johannesson, we wanted to find precisely out if we can identify those two different methylation pathways. So those of within and transgenerational plasticity we would call inducible, and those that would be the result of random epimutations we would call, because we don't have any better word for it, stable. But again, bear in mind, they are of course stable at the range of some numbers of years, but not of course forever. They are probably less stable than normal genetic mutations. And the experimental design, this is now a rather heavy slide. I tried to walk you through that in a bit more detail. So again, we are looking at those three field populations, one from almost complete ocean salinity at 33 PSU, one mid salinity that would be Kiel, and one low salinity that would be in the middle of the Baltic Sea. And we breed mid salinity originating sticklebacks and expose them either to the almost freshwater conditions over 2 generations or to the much higher salinity levels. And then the important thing is here that we then look at the delta, so at those methylated sites that have changed throughout those 2 generations of induction by a changing salinity, and we compare then this delta to what we find in terms of the methylation patterns in the wild. So we are again looking here at full genome data all the time, combined with full methylome data. And first of all, if we do a gene ontology enrichment analysis, we do see that the divergence pattern in the wild compared to the inducible ones in the lab, compared to those that are not inducible, so stable in the lab, that they do show some meaningful patterns. But most interestingly is that the inducible and the stable ones, they diverge quite strongly. So already pointing to the fact that the 2 information pathways of methylation change, as we also can call them, that they are really something different, and they are also attacking different sites on the genome. Then let's come to another very important change that I've showed you in the first half of my talk. That's ocean acidification. That's a simple dissolution of the excess PCO2 that we are producing every day on the temporal scale of weeks with the ocean surface waters. And you've already seen that owing to additional input of CO2 by microbial respiration, which is ultimately a result of too much nutrient influx of the process of eutrophication, we have quite high PCO2 values, which should be a problem for calcifiers, in particular juvenile mussels. So they have to form 2 days after settlements this very delicate shells sitting actually sometimes in the water when they would like to settle. So summer would be between day 200 to 230 of the year. You see it here on the left-hand side when the highest cohorts are coming. So they would like to settle and form their shells when actually the PCO2 conditions are really bad. But somehow they do manage. Now the question was to look at the phenotypic level. So do we see a difference in terms of the local adaptation of mussels that do not regularly experience these very bad PCO2 conditions versus the mussels in Kiel Fjord, or the Baltic Sea mussels, where our hypothesis is they would be able to calcify. So the comparative side was just on the other side of here, the German, this would be Germany, on the other side in the North Sea over here. Yeah, this is the Baltic Sea, this is the North Sea, where we have much more benign and much nicer pH values that are also not fluctuating that much. compared to the Kiel Fjord, the Baltic Sea. So Sylt would be North Atlantic, basically, North Sea, and this is the Baltic Sea. And very nicely, Frank Melzner and his group, so that was Ludger Thomsen, a PhD student of his, they were able to show that in the Baltic Sea, they're just kind of the jacks of all trades, so they can survive under the control PCO2 conditions and at the really heavily acidified conditions. So this would be not even the ocean of 2100. This would be 2.5 times the ocean of 2100, the global open ocean. Whereas in the North Sea, they barely survive. And interestingly, among those Baltic Sea families, there was also a very strong difference in terms of which family was able to successfully settle over the long term. So there seems to be also some within population variation that evolution can, of course, work with if the PCO2 situation in the Baltic Sea would be deteriorating even further. A final selection pressure I would like to present to you that can be of relevance is overfishing. We now know that overfishing can elicit very strong selection pressures that will lead, provided that there are the appropriate heritabilities, to very quick decreases of the age and the length of maturity, for example. So which we also call fisheries-induced selection. And here we also do think that the Baltic Sea, owing to its closeness and owing to the fish population sitting in a pocket, so they really have to evolve in situ. They cannot migrate, they cannot escape that situation. So we do believe that we witness at the phenotypic level one of the strongest and fastest evolutionary responses that I'm at least aware of. So here in this diagram, please look at the red circles. So this is the length at maturity over the past 25 years. And you see that it decreased, and that's true for both males and females, it decreased from about 40 centimeters to about 20 centimeters. So that's the linear dimension. That basically means that a fish that today becomes mature weighs 8 times less body mass than it used to have 25 years ago. And there are, of course, a lot of factors that can lead to those results. Also, less food. You see here that some of the fish are really heavily starving. So this is not how a cod is supposed to look like. It should look like this, with a really fat belly. But be as it may, so one of the ongoing projects we have right now, funded by the German Science Foundation, is that we have a data set based partially on otoliths, partially on fin clips, going back to the 1990s, where we would like to look at the full genome sequences to see whether we are able to find the smoking gun here, to find temporal changes in allele frequency over time that would pinpoint to some maturity related genes. You also may have noted that we have here the length at maturity and not the age of maturity. This is because the age reading from the otoliths in those cods is very difficult, but we now have figured out a way to do that. So we will also be able to soon be able to plot that graph here as it should be for other world regions, meaning the age at maturity. Yeah, finally, being a brackish water area means that such an ocean basin is always transient. So to my knowledge, and I think that's also agreed upon by geologists, brackish sea areas are always ephemeral if you look really at geological timescales. So we either do have fresh water or we have full ocean salinity. Nevertheless, it seems to be the case that the 5,000 to 6,000 years that the Baltic has now existed after the last glaciation as brackish sea area were long enough to result in some speciations already. So one of the novel species that at the same time is also very interesting because it's an asexual speciation is the Fucus radicans, described by Kerstin Johannesson and group. So that's a bladder wrack that is not becoming fertile anymore and that is simply proliferating in the central and northern Baltic by vegetative fragmentation. And its mother species, if you will, the ancestral species would be Fucus vesiculosus. That's still occurring, in the Kiel area, for example. So partially they also coexist the novel asexual species and the ancestral original, also sexually reproducing species. If this seems to be, if you don't agree that something asexual can be called a species, I mean, that's of course an open debate, which you would have to conduct with Ricardo and Kerstin, not with me. I'm just referring to that very interesting example. Another example where actually speciation has taken place is in flounder. And this is actually triggered most probably by the very bad oxygen situation in the deep basins of the Baltic Sea. I forgot to mention that the Baltic is also a very drastic example for ocean deoxygenation. The situation here is exacerbated by the vertical salinity stratification, so lighter, a little bit fresher water layers are sitting like a lid on the deeper, more saline bottom waters, and they are preventing almost all exchange with the surface, even in winter. And so in the central and northern basin, the flounder that's originally a fish that is a benthic spawner has developed into another reproductive mode, which has pelagic eggs. So eggs that are floating and are therefore not sinking into the bottom layers where they simply would die owing to oxygen deficiency. So this is, of course, very interesting, and they really figured out that the gene pools have been completely separated. So if you will, the species pair now perfectly fulfills Meyer's biological species criterion by having two sympatric populations whose gene pools are absolutely not mixing anymore, which is clear because they have a completely different reproductive mode and reproductive timing now. Yeah, to sum up, so the Baltic Sea is an ecological and a time machine and a natural evolution lab. So several factors like temperature, PCO2, aka ocean acidification, oxygen, which I forgot to mention initially, but you here see the dramatic diagram. So the black areas here, that's about 100 years ago, was the extent of bottom oxygen deficiency. The red one is the extent in the past decade, so a 10-fold increase, or also the standing gradient in salinity that lends itself to space for time design. So all of these factors make it a very, very interesting evolutionary time machine. So let me sum up first the aspect of the Baltic Sea generally as a nice comparative system for coastal seas. So I hope I've shown you that in summary, if you aggregate all the different pressures, the Baltic is probably one of the most heavily influenced world regions in the world. So it perfectly fits a complex, high pressure complex management framework, complex management because we have 9 different countries forming its watershed. But fortunately, the Baltic Sea has been studied for about 100 years very intensely, which gives us scientific collaboration and long-term data series, which are the condition for science-based management. And together with the political will of various organizations, most importantly here to name is the Helsinki Commission. There were some management successes that can be role models for other world regions. However, the elephant in the room is climate change that's increasingly jeopardizing the success of those management successes. And if you look at the evolution aspect, so here we have a paradox. we have generally lower genetic diversity of the populations sitting in the Baltic. We also probably have some further decrease of general genetic diversity by strong selection pressures and genetic hitchhiking, although the evidence for that is still not conclusive. Nevertheless, we do see strong adaptation wherever we look for the very drastic conditions in the Baltic, which offer kind of a nugget of hope, I would say. So some of the species, such as mussels, but also cod, where I didn't show you the results. So the cod in general seems to be absolutely resilient even to those bad PCO2 values. If we are doing the experiments with Baltic Sea cod, if we do the same experiments with North Atlantic cod, we do see very drastic negative effects of ocean acidification. The Baltic has very strong gradients. Most pronounced is the salinity gradient, where we can do space for time experimental designs, combining manipulative experiments with looking at the patterns of standing genetic variation in the wild. Yeah, this I've already said, actually. So many Baltic species are adapted not just to the contrast, but also to the fluctuations in conditions. I've tried to show you a case where also epigenetic changes seem to contribute to salinity adaptation, and also the large degree of clonality lend some of the Baltic macrophytes to studies where we look at the role of somatic mutations, how they proliferate within clones, and how they can also maybe contribute to adaptive evolution. And with this, I'm at my end. I thank you for the attention. I'm very happy to take on questions. And I will now stop the screen so that I actually can show you, can see you.
Thank you very much for that very interesting talk. I would like to move us into the Q&A session, the question and answer session. If anyone has a question, you can either use the blue raise your hand function in the participants tab, and I can call on you, or feel free to put your question in the chat and I can go ahead and read it. And I see we have a question straight away from Ileana. Do you want to take us on this journey?
Thorsten, thank you for a really interesting talk. I love all of those examples of how many different ways the individual species seem to be adapting to this pretty rapidly changing environment. So in some ways, it seems very hopeful, but I'm wondering about what doesn't seem to be happening as well as how the interactions evolve amongst those species. Is this just, like, so where does this leave us? So if the individual species can sort of deal with it, but the interactions are lagging behind, are we still predicting basically ecosystem collapse as a result?
Yeah, ecosystem collapse. I mean, for sure, we have pretty drastic ecosystem remodelings at the moment all the time. What I didn't mention is invasive species. So because the Baltic Sea is considered species poor, because it's such a stressful environment, so only a subset of the oceanic species can enter there. So any influx of invasive species messes everything totally up, and that's what we're seeing right now. For example, there's the round goby coming in. That's a small fish from the Ponto-Caspian area that's very tolerant to very low salinities, but can also go up to normal ocean salinities. It's basically eating up all bivalves, totally. So where you had mussel beds before, they're just gone. It's just one example where you have this situation that the ecosystem configuration is changing really very, very rapidly. So your question is really a good one. At the moment, we've more focused on a few model species, how they cope. But what we see, let's say, from the ecological angle is that the communities are super rapidly shifting. Also, the major commercial fish are probably doomed to go extinct. So just by warming, the cod will probably not prevail. And then a top predator is just lacking. So that's really the top predator fish. And it remains to be seen if sea bass and sea bream are really entering, at least the western part. So they're now starting to come in. So everything is really in fast forward. Everything is just going super rapidly at the moment. The reconfiguration and also the changing of the major players.
Great. Our next question will be from Eric Crandall. Do you want to go ahead and unmute yourself and ask the question?
Hi. Yeah, just a really basic evolution question is, it's pretty clear that there's speciation happening along the environmental gradients there, but is there any evidence, or I'm just not familiar with the system, is there any currents there that might actually act as allopatric barriers, like things that are just physically blocking larval dispersal, or do you think it's entirely environmentally driven?
Those examples I gave, I think they are mostly environmental driven. We have very, very good current models. And I think in terms of surface currents, so basically propagules are circulating in the Baltic on a scale of months. They are also very good circulation model in terms of devising marine protected areas, but it's just the spatial scale is so small. I think that we in most cases can dismiss really strong barriers like Point Conception or something like that we do see in other oceanic systems.
Great. I'm going to encourage graduate students to also ask some questions, and I'll ask one in the meantime that I have. I'm a microbiologist by training, so I'm very curious to understand how microbial adaptation may be contributing in the Baltic Sea. So I've seen a lot of really great studies that have looked at sediment and tried to understand how the microbes in the water have changed through time. But I'm really curious to know about whether or not somebody has looked at microbial adaptation within the species that you're looking at. So we know salinity contributes to microbial community structures quite strongly, and in fact, maybe the strongest factor on the planet. And so do you see evidence of adaptation or of benefits from having different microbes in some of the eukaryotic systems that you're looking at?
Yeah, I'm sorry, we haven't looked at that yet. So we don't know. I know it's a very interesting hypothesis to follow, but at the moment we simply don't know how that's going to play out. I mean, what's clear is that salinity is the major decisive factor which microbial communities you have, either associated with hosts or free living. That's clear. Yeah. No, I'm sorry, I cannot answer this.
Well, with that, I'll go ahead and close today's session. Thank you so much for your wonderful talk today, Thorsten. It was fantastic, very compelling from my perspective. The next talk will be likely in February of spring 21, so please watch your email and be prepared for the next announcement. Everybody be healthy, be well, and take care.